Good afternoon, students. In today's lecture, we are going to discuss hybridization program in the sorghum. In the last lecture, we have discussed the hybridization program in cotton, and in today's lecture, we are going to discuss the hybridization program in the sorghum. Okay, today's topic for the discussion is the hybridization program in the sorghum. Here, the first of all, introduction to the sorghum, or popularly we call the sorghum as the jawar. Okay, the sorghum it comes fifth after wheat, maize, rice, and barley in both area and production. Okay, sorghum comes fifth after wheat, maize, rice, and the barley in both area and its production. Okay, the jawar it is the crop, the grains of the jawar, that is these are used as a human feed, and also the plant it is used as the fodder. Okay, the sorghum plant, the grains of the sorghum, these are used as the human feed, and also the sorghum plant it is used as the fodder. Okay, it is one of the most important food crops in the semi arid and the tropics of the India, Africa, Australia, Argentina, and Mexico. Okay, it is one of the most important food crop in the semi arid and the tropical regions of India, Africa, Australia, Argentina, and Mexico. In India, it is cultivated on large scale in Maharashtra, Gujarat, Tamil Nadu, Karnataka, Rajasthan, and Madhya Pradesh. Okay, these are the states which are cultivating the sorghum in the higher level. Okay, on the large scale. Okay, on the large scale in India, it is cultivated in the states like the Maharashtra, Gujarat, Tamil Nadu, Karnataka, Rajasthan, and Madhya Pradesh. Okay, and the sorghum it is believed to be originated in the Ethiopia and South Asia. Okay, the sorghum it is believed to be originated in Ethiopia and South Asia. The progenitor of the sorghum bicolor. Okay, sorghum bicolor, that is the species of the sorghum which is widely cultivated on the commercial scale. Okay, that is the botanical name of the sorghum, sorghum bicolor, and the sorghum bicolor it is belonging to the family Poaceae. Okay, it is belonging to the family Poaceae. Okay, it is the commercial cultivated species of the sorghum, sorghum bicolor, and the progenitor of this species is believed to be the sorghum arundanaceum. Okay, sorghum arundanaceum, that is believed to be the progenitor of the sorghum bicolor. Sorghum it is believed to be originated in Ethiopia and South Asia. Okay, that is about the uh, general account of the crop sorghum. And here, the most of all, most importantly, we are plant breeders. These are mainly concerned with the floral biology. First of all, in this one, we are going to discuss the floral biology, or the structure of the flower in the sorghum. Okay, here the inflorescence of the sorghum, here you can see it is the panicle. Okay, and the panicle consists of spikelet in pairs. Okay, sometimes these spikelet, the three spikelets may be present. Okay, here you can see that is there is a presence of the spikelets. Here you can see the double spikelet unit. Okay, that means the spikelets these are present in the pair. But here you can see the triple spikelet. Okay, there may be the three spikelets may be also present in the panicle. Okay, here the you can see here that is it is the sessile spikelet. The sessile spikelet it is hermaphrodite. Hermaphrodite means it is bisexual. Here both the male reproductive organ, androecium and gynoecium, are present, and that's why this sessile spikelet, as it is hermaphrodite, it is referred as the perfect spikelet. While the another one, that is the pedicellate spikelet, in these spikelets there is a presence of the it is a sterile spikelet. It is called as the sterile spikelet because it is having only the male reproductive organs, androecium, and that's why the pedicellate spikelet it is referred as the staminate spikelet. Okay, whenever the spikelets are present in pair, the one is a sessile spikelet and the another one it is the pedicellate spikelet. Sessile spikelet, it is the hermaphrodite, it is bisexual, and that's why it is referred as the perfect spikelet. And the another spikelet which is pedicellate, it is a sterile and it is staminate spikelet. Okay, it is staminate spikelet. Here you can see the uh, whenever, that is the spikelets, these are present in three. Whenever the triple spikelets are present, the of these the two spikelets, these are pedicellate spikelet, that means these are the staminate spikelet, and there is a presence of one, only one sessile perfect spikelet. Okay, whenever the triple spikelets are present, of these the two spikelets, these are staminate spikelet, that means these are pedicellate spikelet, and only one spikelet is a sessile which is perfect spikelet, or we can say which is bisexual. Okay, that is about the panicle consisting of the spikelets. Here you can see the sessile spikelet, it consists of inner and the outer glooms. Okay, the sessile spikelet here, it is consisting of the inner and the outer glooms, including the two flowers. The upper one it is perfect and the lower one it is reduced. Okay, here you can see the sessile, the perfect spikelet, it is having the two flowers. Okay, it consists of inner and outer glooms. Okay, and in between the inner and the outer glooms, you can see there is a presence of the two flowers, of which the upper one it is perfect and the lower one it is reduced. Okay, here you can see the structure of the perfect flower. Okay, perfect flower, or we can say the bisexual flower. Okay, the perfect, it is called as the perfect flower because it is having all the floral whorls. Okay, it is having male reproductive organ androecium and female reproductive organ gynoecium. Here you can see the structure of the perfect spikelet. Okay, here the perfect flower has a thin, narrow, hairy lemon. Okay, here you can see this perfect spikelet, it is having the thin, narrow, sterile, hairy lemma, and along with the lemma there is a presence of, here you can see it is the sterile lemma. Okay, thin, narrow, hairy lemma it is present, and it is also having along with this one small pally. Okay, here you can see there is a presence of the small pally, and this pally includes here the stamens. Okay, it includes the three stamens and the two lodicules. Okay, here you can see here the two lodicules are present. Okay, that means the pally it is including the three stamens and the two lodicules. Here you can see the presence of the lodicules. Okay, and also it is having the bifurcated feathery brush like stigma. Okay, here you can see the bifurcated bifurcated feathery brush like stigma. Okay, this is the structure of the perfect or the bisexual flower in the jawar panicles. Okay, I am repeating this one, that is the perfect flower, it has thin, narrow, hairy lemma. Okay, here you can see the presence of the thin, narrow, hairy lemma. Along with this, there is a presence of the pally. Okay, this green colored here you can see the presence of the pally, and that pally it is including the stamens. Then here you can see these are the two lodicals, and here you can see the bifurcated feathery or the brush like stigma. Okay, the pedicellate flower, pedicellate, or we can say the staminate flower, the staminate flower, it is without the pally and the ovary. Okay, it is not having the female reproductive organ gynoecium. That's why the pedicellate flower it is said to be the it is said to be the staminate flower. Okay, the pedicellate flower it is without the pally and the ovary, without female reproductive organ gynoecium. Okay, then the flowering it generally occurs prior to the sunrise and extended up to the midday. Okay, the flowering, it occurs prior to the sunrise and extended up to the midday. Okay, the blooming starts from the tip of the panicle in the downward direction. Okay, here you can see the flowering, it occurs prior to sunrise and it is extended up to the midday. The blooming, it starts from the tip of the panicle and in the downward direction. Okay, the stigma, it is receptive before flowering and remains receptive for about 6 to 8 days. Okay, the stigma is receptive before the flower opens and remains receptive for about 6 to 8 days. The pollens, these are viable for only few hours. Okay, pollens are viable for only few hours, and the fertilization process, it is get accomplished within 2 to 4 hours of the pollination. Okay, as the pollens are viable, that's why the pollination and the fertilization process, it is accomplished in the 2 to 4 hours after the pollination. Okay, then the sorghum is a normally self-pollinated crop. Jawar, it is a normally self-pollinated crop, but the stigmas exposed before dehiscence lead to the 6 to 30% of the cross pollination. Okay, that is the jawar, it is referred as the self pollinated crop. But what happens, the sometimes the stigma becomes receptive before the anther dehisces. Okay, before the anther dehisces, and that's why that stigma receives the pollen grains from the other crops. Okay, and that leads to 6 to 30% cross pollination. Okay, the glumes open due to swelling of lodicules and another come out of the stigma. Okay, the glumes open due to the swelling of lodicules and another come out of stigma. Okay, then the stigma remains receptive for about 88 to 16 days after blooming. Okay, the stigma remains receptive for about 8 to 16 days after blooming or after the opening of the flower. Okay, here you can see how actually the emasculation and the pollination process is carried out. Okay, how actually the emasculation and the pollination process is carried out. Here the different methods of the emasculations are used in the sorghum, in the breeding program of the sorghum. The first one, here we are going to discuss the hand emasculation. Hand emasculation, only the part of the peduncle is emasculated. Okay, in the emasculation, only the part of the peduncle, it is emasculated, and flowered tips and the lower branches are removed by the clipping. Okay, in the hand emasculation, how exactly the hand emasculation in the sorghum is carried out, only the part of the peduncle is emasculated and the flowered tips and the lower branches, these are removed by the clipping. About 50 florets that would flower on the next day are selected. Okay, that is about 50 florets, means about 50 perfect flowers, that is these are generally selected which are likely to open on the next day. Okay, about 50 florets that would flower on the next day, these are selected and these are emasculated, and then after the emasculation, these are covered up with the suitable paper bag. Okay, that is just to avoid the random cross pollination, the bagging is carried out after the emasculation. Okay, we have discussed in detail regarding the emasculation and the bagging process. Okay, emasculation means what? The removal of stamens or the removal of anthers, or killing the pollen grains without disturbing the female reproductive organ gynosium is called as the emasculation. Okay, and emasculation is essential in the female parent just to check the random pollination. Okay, whether it is self pollination or the cross pollination. Okay, about 50 florets that would flower on the next day, these are selected and then these are emasculated, and after the completion of emasculation process, immediately these emasculated flowers, these are covered up with the suitable paper bag just to avoid the random pollination. Okay, then another method which is used for the emasculation in the sorghum, it is the hot water treatment. Okay, hot water treatment. In this method, the sorghum in this method, the sorghum head is immersed in the water at the 45 degrees centigrade to the 48 degrees centigrade for 10 minutes without injury to the stigma. Okay, in this method, the sorghum head is immersed in the water at 45 degrees centigrade to the 48 degrees centigrade for about 10 minutes so that the stigma will not get injured. The most important thing here, the temperature of the water and the duration. Okay, these are the two most important things which the plant breeder have to take in. Plant breeder have to consider. Okay, and the precaution should be taken that the stigma will not get injured due to these treatments. Okay, emasculation and pollination. The first method of emasculation which is used commonly in the jawar, these are hand emasculation. Then the second one, it is the hot water method. Okay, then another method is used and that is called as the plastic bag emasculation. Florets open and then the anthers emerges, but they will not shed the pollens. Okay, the anthers will not shed the pollens due to the high humidity inside the bag. The anthers are knocked free of head by tapping. Okay, just by tapping, the anthers are tapped with the help of the fingers, and by tapping, then the pollens will be free. Okay, in this method the some selfing occurs. Okay, some selfing occurs or the some self pollination may occur. Therefore the marker genes are needed to identify the plants arising from the selfed seeds. Okay, that is the another method which is used in the emasculation of the sorghum. In this method, the sorghum panicle is covered with the plastic bag. This will create the high humidity inside the bag. Under such humidity, the florets open, the anthers emerges, but shed no pollen. Then the anthers are knocked free of head by tapping. Okay, in this method some selfing occurs and therefore the marker genes are needed to identify the plants arising from the selfed seed. Okay, then after the emasculation the pollination is carried out. Okay, in the pollination the fresh and viable pollen grains from the selected male parents are collected and then these are placed, these are kept on the receptive stigma of the female parent. Okay, here that is how the pollination is carried out. Here the pollination it is done on the next day between 9 to 10 a.m. when the all flowers come to bloom. Okay, the pollination it is carried out on the next day between 9 to 10 a.m. when the all flowers come to bloom. Okay, then generally the heads or the generally the heads that is inserting and shading the head in the bag collect the pollen. Okay, just the heads of the sorghum flower, that is these are shaken and then by just shaking their pollen grains will be collected in the bags. Okay, then the another technique is the clipping the heads early in the morning and placed in the boxes or the flower pots kept in the protected place. Flower pots kept in protected place. Then the collected pollen grains, these are dusted over the exposed stigma or the pollens producing the heads and brushed over the emasculated head. Okay, the collected pollens, these are dusted over the exposed stigma or the pollens producing the heads and brushed over the emasculated. Okay, that is how actually the pollination is carried out in the sorghum. Here in this method you can clearly see how exactly the emasculation is carried out. Okay, here you can see that is how the emasculation is carried out, how the anthers and the stamens, male reproductive organs, these are removed. This is in the photograph of A, B, C, and the D. Okay, and here you can see the clipping the head of the sorghum. Okay, that is the another method which is used for the emasculation. Okay, that is inside the bag. There will be the high humidity, and that's why the flower will open, the anthers will not shed the pollen grains. In that one, these anthers, that is these are knocked just by tapping, and that is how the emasculation is carried out. Okay, this is the first photograph which is showing the emasculation process in the sorghum. Okay, and in this one you can see the pollination, that is the bagging, it is carried out in the male flowers. Okay, the male flowers, that is these are bagged in the suitable size of the paper bag, and so that after the opening of the flower and after the dehiscence of the stamens, the pollen grains will be ultimately collected in the bags, and these fresh and viable pollen grains, these are then used for the pollination process, or these are then brushed, these are then kept on the receptive stigma of the female flower with the help of the hairbrush. Okay, that is the emasculation and the pollination process in the sorghum. Then what are the breeding objectives of the sorghum? Okay, the breeding objectives in the sorghum, the first one, it is the high yield fertilizer response. Okay, the first one, it is, sorry, the first one here you can see the high yield. Okay, the variety which is produced, the new variety which is produced by the breeding program in the sorghum should be having the high yield. Okay, and it should be fertilizer responsive. Okay, then another, here you can see the wide environmental adaptiveness, the variety should be get adapted to the different environmental conditions. Okay, so that it may be cultivated in the different regions. Then the disease and the insect resistance. Then non-lodging. Non-lodging means what is mean by lodging? That is due to the excessive growth of the sorghum, the lower internodal region, it will, or the lower stem region, it will become very weak, and that's why, due to which the plant, it tends to fall on the ground. That phenomenon, it is called as the lodging. Okay, here the breeding, another breeding objective in the sorghum, it is the non-lodging. Then the appropriate time to maturity. Okay, it should be maturated in the appropriate time. Then good threshability. Then general attractiveness. Then another breeding objective, the head, it should not be too compact or not too grassy. Then the head erect rather than the curved. Then good tillering with the heads on all comes and maturing the same time. Okay, good tillering means the branches. Okay, and the heads on all culms, the stem in the sorghum, it is referred as the culm. Okay, with the heads on all culms, that means the variety which is released by the breeding, it should be having the good branches, and each of the branch, there should be the formation or the development of the head or the inflorescence, and all these heads should mature at the same time. Okay, then good seed set. Then the breeding objective is the seed size and the number. Okay, these are the different breeding objectives on the sorghum. Then the breeding procedures. Okay, different methods which are used in the sorghum for breeding. Here, the first one is the pure line selection. Pure line selection, in this method, superior land races are selected from the local uniform variety. Okay, that means the first of all, the selection it is carried out from the local uniform variety. Okay, in this method, the superior land races or the local varieties are selected from the local uniform variety. Okay, and then these are grown as the plant progenies in the next year. Okay, the progenies of these plants, these are grown in the next year. The uniform superior progenies, these are harvested, these are bulked as the improved strain for the further evaluation. And then in varietal evaluation, if the improved strain shows the superiority over the existing variety, then it is released for the commercial cultivation on the large scale. Okay, we have all discussed in detail how actually the pure line selection is carried out. Okay, how actually the pure line selection is carried out. In the pure line selection, when it is practiced in the sorghum, the first of all, the selection is carried out from the local variety. Okay, the selection is carried out from the local variety, and then their progenies are grown. Okay, later on, the uniform progenies or the plants with the uniform characters, these are selected, these are bulked together, their seeds are bulked together, forming the improved strain. Okay, then the manual trials and the district yield trials are carried out. Okay, and later on, the improved strain which performs the better than that of the existing variety, it is released for the commercial cultivation on the large scale. Okay, here you can see the different varieties which are released by using this method. Okay, for example, M35-1, then Cell 3, Yashda, and the Mauli. These are the different varieties which are released by using the pure line selection method. Okay, then the second method, it is the pedigree method. Pedigree method, this method consists of hybridization. Okay, pedigree method. This method consists of hybridization between desirable complementary parental lines. Okay, desirable complementary parental lines, followed by the selection of superior plants in the segregating generation till the homozygosity is achieved. For example, it is carried out up to the F5 to the F6 generation. Okay, this method consists of hybridization between the desirable complementary parental lines, followed by the selection of superior plants, and in the segregating generation till the homozygosity is achieved. That is up to the F5 to the F6 generation. Okay, then the selected plants are bagged to prevent out the crossing. Okay, the selected plants, these are bagged to prevent out the crossing. The varieties which are developed by using the pedigree method are SPV86. Here you can see the parental lines are R24 and the R16. Okay, and another variety which is released by using this method is the SPV 504, and that variety is also referred as the Swati, and the parents are SPV 86 and M35 1. Then another variety, it is the CSV 15R, and the parental lines are SPV 475 and SPV 462. Okay, this method it consists of hybridization between the desirable complementary parental lines, followed by the selection of superior plants in the segregating generation till the homozygosity is achieved, that is up to the F5 and the F6 generation. And the selected plants, these are bagged to prevent out the cross pollination. Okay, then The another method which is used in the sorghum, it is the backcross breeding. In the backcross breeding, it is used to transfer one or few inherited traits from the donor to the another desirable genotype. Okay, and that is referred as the recipient parent. Okay, backcross method, it is generally used to transfer the one or more character from the one plant which is referred as the donor plant to the another plant which is referred as the recipient parent. Okay, and generally this method, this backcross breeding method, it is used to used in the plants to increase the or to improve the resistance to diseases like the grain mold, downy mildew, rust, smut, and also the resistance to the different insect pests like midge, shoot fly, stem borer, and it could be introduced in the desirable strain by the back cross breeding method. Okay, by using this method, the resistance to diseases like the grain mold, downy mildew, rust, smut, and resistance to the insect pest like the midge, shoot fly, and the stem borer, it could be introduced in the desirable strain by using this method. Okay, similarly, the cytoplasmic genetic mull sterility, it could also be introduced by using this method. Okay, similarly, the cytoplasmic genetic male sterility could also be introduced by this method. Then the another method which is used in the sorghum, it is the hybrid breeding. Okay, hybrid breeding, here the seeds of the hybrid sorghum, these are produced using cytoplasmic genetic male sterility. Okay, seeds of the hybrid sorghum, these are produced using cytoplasmic genetic male sterility, typically known as the A, B, and the R line system. Okay, seeds of the hybrid sorghum, these are produced by using the ABR line system. Okay, here the combined kafir 60 was initial ms line which is used in the hybrid seed production. Okay, combined kafir 60 ms was the initial ms line which is used in the hybrid seed production. Okay, that is used as a mother strain line which is used in the hybrid seed production. Okay, then line A has the sterile cytoplasm and line B has the fertile cytoplasm. The only difference in between the line A and the line B is that the line A has the sterile cytoplasm while the line B has the fertile cytoplasm. The new hybrids can be developed by improving the performance of A and R lines or both by the back cross breeding method. Okay, the new hybrids in the sorghum can be developed by improving the performance of A and R lines or both by the back cross breeding method. Every year, every year, the crossing program of restorers, which we referred it as a R line. Every year, the crossing program of restorers with the promising male sterile line is undertaken and the hybrids are tested for the performance. If the performance of the particular hybrid found superior, it is multiplied on the large scale for the commercial cultivation. Okay, every year the crossing program of the restorer, that means the R line, with the promising male sterile lines is undertaken and the hybrids are tested for the performance, and if the performance of the particular hybrid found superior, it is multiplied on the larger scale for the commercial cultivation. Here you can see the some of the popular hybrids, these are released by using this method are CSH1, CSH5, CSH12, CSH15, and CSH16. Okay, that is about the hybrid breeding in the sorghum. Okay, and now the international and national programs. Okay, the International Crop Research Institute of Semi-Arid Tropics, which is in short form is called as the ICRISAT. Okay, International Crop Research Institute of Semi-Arid Tropics. It was established in the 1972 at the Hyderabad. Okay, International Crop Research Institute of Semi-Arid Tropics. ICRISAT, it was established in the 1972 at the Hyderabad. The sorghum variety ICSV112, or it is referred as the SPV475. It was released as the CSV13 for the general cultivation in India. Okay, ICRISAT, it is working with the collaboration of NRCS, okay, National Research Center on Sorghum at the national level at Solapur for the sacristy area. Okay, ICRISAT, it is working with the collaboration of NRCS, National Research Center on the Sorghum at the national level at Solapur for the sacristy area. Okay, then the organized sorghum improvement program in India, it was started in the 1962 when Indian Council of Agricultural Research initiated accelerated hybrid improvement project. Okay, organized sorghum improvement program in India started in the 1962 when ICAR, Indian Council of Agricultural Research, initiated accelerated hybrid improvement project. Then the All India Sorghum Improvement Project on the sorghum, it was established in the 1969 at the Hyderabad, involving Indian Council of. Okay, All India Sorghum Improvement Project on Sorghum was established in the 1969 at the Hyderabad, involving Indian Council of Agricultural Research and state agricultural universities. In Maharashtra, the project officers are doing research at the following universities on the various aspects of the sorghum. In this one, the first one is the Mahatma Phule Krishi Vidyapit, Rahuri. Then second one, it is the Punjab Rao Deshmukh Krishi Vidyapit, Marathwada Krishi Vidyapit, Parphani.